All right. Thank you and welcome to everyone for joining me today for the digital health webinar on the predictive diagnosis of ocular disease. This webinar is brought to you by UCLA's Technology Development Group. TDG serves as UCLA's campus-wide gateway to innovation, research, and entrepreneurship. It's my privilege to be your host today. My name is Joel Cayley. I am the Business Development Officer in TDG's Technology Transfer Practice focused on digital health. My guests today are Dr. Aran Halperin, Dr. Srinivas Sada, and Dr. Ali Hatamazidai. Dr. Halperin is professor in UCLA's Department of Computer Science, Computational Medicine, Anesthesiology, and Human Genetics. He's also the head of the AI in Medicine program, the Associate Director of Informatics in the Institute of Precision Health, and the co-director of Computational Genomics Summer Institute at UCLA. Dr. Halperin received his PhD in computer science from Tel Aviv University. Prior to his current position, he held faculty and postdoctoral positions at the University of California, Berkeley, the International Computer Science Institute at Berkeley, Princeton University, and Tel Aviv University. Dr. Halperin's research focuses on the development of computational methods on medical and biological data in the context of human diseases. He has published over 150 peer-reviewed articles across different disciplines such as computational biology, computational medicine, human genetics, and machine learning. He received various honors from academic achievements, including the Rothschild Fellowship, the Technion Julidan Prize, the Krill Prize, and most recently, he was elected to be an ISCB Fellow. Dr. Srinivas Sada is the President and Chief Scientific Officer of the Doheny Eye Institute, the Stephen J. Reinhold, Arnold, and Mabel Beckman Endowed Chair, and Professor of Ophthalmology at the UCLA David Geffen School of Medicine. He received his MD from Johns Hopkins University, where he also completed ophthalmology residency and neuro-ophthalmology and medical retina fellowships at the Wilmer Eye Institute. Dr. Sada's major research interests include automated retinal image analysis, retinal substructure assessment, and advanced retinal imaging technologies. He has more than 380 peer-reviewed publications and 13 book chapters and has given over 350 presentations worldwide. Dr. Sada also serves as an editorial board member of Ophthalmic Surgery, Lasers and Imaging, Retina, Ophthalmology, Retina, and Ophthalmology. He is also an editor of the 5th edition of Ryan's Retina textbook. Among Dr. Sada's awards and honors are a Research to Prevent Blindness Physician Scientist Award, the Macular Society Paul Henkin Lecture and Award, and the American Society of Retina Specialists Young Investigator Award. He's also received the Silver Fellow designation from the Association for Research in Vision and Ophthalmology. He has been named the Best Doctors of America list for several consecutive years. Dr. Ali Hatamazidai is a research scientist at NVIDIA. His research interest lies at the intersection of computer vision, natural language processing, and machine learning for medical applications. He's also received his PhD and Master's in Computer Scientist from UCLA. He is the recipient of numerous awards, including the 2018 UCLA Henry Samueli School of Engineering and Applied Science Award, the Best Paper Award for 2019-2020 biennium of the CMBBE, and NVIDIA Best Paper Award for the MICC AI Workshop on Deep Learning for Medical Image Analysis. Gentlemen, thank you for joining me today to share all of your work with our audience. For the audience, Dr. Sada is in clinic today, and so I expect him to be joining us when he's, as he's juggling his workload, he will be joining us to answer questions about his research. So I'd like to start by having Dr. Halperin tell us about his work with Dr. Sada on automated biomarker prediction.
Okay, thank you. I'm going to start by sharing the screen. Can you see the screen?
That looks great. Thank you.
Okay, great. Okay, so thank you, Joe, for the introduction. And this work is a joint work with Dr. Sada that is going to join us from the from the clinic. And basically what I'm coming more from the engineering side, and Dr. Sada comes from the clinical side. And basically for most of the projects that I've done in the past 20 years, I think they're usually successful when you have people coming from the different sides and seeing how things should work from both sides, or sometimes more than two sides. So what I'm going to talk today about is some deep learning technique that we developed related to age-related macular degeneration using imaging technique, which is called OCT. So just for this is going to be a very short presentation, and I'm going to just basically give kind of an overview first about deep learning and about the motivation for this project. So I'm sure everyone heard about deep learning, and really the promise of deep learning is that it's one of the techniques that has seen, I would dare to say, the most success that you've seen in machine learning, or at least in the last 10 years, in terms of applications, not only to medicine, but also to other parts of life. For medicine specifically, there's a lot of work that has been done on imaging, but also on other parts of medicine, that physiological waveforms and electronic health records. And in principle, we could, there's many tasks that we see that the computer using these deep learning techniques can potentially do a work in a much more automated way than what the clinicians are doing, often in par in terms of the accuracy of the diagnosis, and so, which is basically something that we could use to reduce the load on the clinicians. There's many applications. Just to give you an idea, for those of you who haven't seen, basically in deep learning, we can think of a deep learning architecture or a model as a set of small computing machines. Each of these can be called is typically called a neuron, so that's why it's a neural network. And it does a very small computation. So the basic idea is that each of these neurons is doing a very small computation. You start, let's say, with a picture of a dog or a cat, and you want to know whether it's a dog or a cat. So each of these neurons is doing some calculation that basically is passed to the next layer, and so on. And then in the end of the calculation, we get the idea that this picture specifically is a picture of a dog. If we had a cat, then it basically give us a cat. And so this is kind of the, obviously in a very simplified way, what we have for in a typical neural network or deep learning. So it's called deep because in many cases we're looking at many layers of this neural network. In this case, it's only one hidden layer and then just the input and the output. But you can imagine that this could be 50 layers or even more. Okay, so that's deep learning. And the problem with, at least one of the limitations of deep learning, is that it requires quite a lot of data. And the reason is to require such a large amount of data is because usually when you look at neural networks, and especially deep ones, again, this is an example of a very simple one, then you need to learn many parameters. So these are just some typical examples from some networks or architect models that are widely used. So the ResNet 50 model has about 23 million parameters. The ResNet 152 model, which is a bigger model, has about 60 million. BERT, which is another neural network that is used for natural language processing, has about 100 million parameters, and GPT-3, 175 billion, and there's even more than that. But you can see that the number of parameters is huge. When you have a large number of parameters, you need large amounts of data. So if we need a large amount of data for training, that basically means that we need to get, if it's images, in the case of what I'm going to present today, you need for every image to know whether this image is for a patient that has, let's say if you're looking at a specific disease, that has the disease, or a patient that doesn't have the disease, so you need some label data. And in medical settings, this is often hard to get. So this label data is hard to get. In other settings, it could be easier. So for example, with the case of a dog or a cat, you could potentially just ask many people to say whether what they see is a cat or a dog. But in medical settings, you have to go to professionals, and it's harder to obtain this. So I'm going to talk about a specific application. So we basically try to work around this limitation in a specific way. And the example that I'm going to give here is that of application to age-related macular degeneration, which is the leading cause of blindness in the US. And it's a disease that basically, if you wait long enough, then all of us are going to get this. But the question is really, if you're starting to show symptoms when you're 50 or when you're 70 and so on. So basically the question is really how fast do you deteriorate, or our eyes are deteriorating, or retina deteriorates, and not whether it's going to deteriorate. It's probably going to happen, but basically the question is what is the pace and what is the rate. And this is again, it's one of the leading causes of blindness, and one of the major ways, or I think the main way of diagnosing it, is using this technology called OCTs, optical coherence tomography, which you can think of this as ultrasound, but basically instead of sound, it's light that gives you a three-dimensional image of the retina, and so what you see here is that, I don't know if you can see my cursor, hopefully you can see that basically what you get are essentially a set of images that are just next to each other, and each of them corresponds to one slice of the retina. So all together, you get a three-dimensional picture of the retina. And when you're looking at the patients, you're looking for some abnormalities, so things that are different from if you have a healthy control. If you're looking at a patient, you want to see something which is different, and then you say, oh, there's some type of problem. Many of these images are images that are looking from the side. So this is kind of this is basically what you see here. You have three different axes. So the images that you see here on C, so you have A, B, and C, are images that are basically crossing sideways the retina, and so you have and so when you look at this, you can see the different layers of the retina, and then again in a healthy patient, you'll see that this is basically very smooth, and in a patient that has AMD, you'll see that there's these kind of bumps that I'll talk about this in a second. So a lot of what's going on in the field is that basically the ophthalmologists are trying to understand what is the process of deterioration of the disease. So you start with a normal retina, and then over time there's some progress in the disease, and you have some early AMD where you have some this is a close-up on these bumps in the image. And this is basically becoming worse and worse until basically there's really this atrophy, which is essentially the it's completely damaged. But on the way, there's a lot of biomarkers that we can basically look at and suggest that the patient is getting worse, and potentially these are predictive, not potentially, these are often predictive of the patient getting worse in the next year or two, so you can know kind of what's going to be the progress of the disease in the next couple of years. This is where we are coming in. We're basically looking at these features that are essentially biomarkers for the progression of the disease. In our study, we looked at 5 specific features that we had access to that data through annotation that came from Dr. Sada's group, that they basically went and manually inspected each of the volumes. So I'm calling them volumes because these are 3-dimensional images. So I'm not going to get into the details of each of them, but these are 5 different features that each of them has these kind of examples of basically, that's an example for an image that has this type of feature. So you can see this kind of circle here, which which is filled with kind of cloud, and here you have some parts that are basically missing. And so in each of them, we have some characteristics that are sometimes easier and sometimes not as easy to find. Sometimes they're only found in one of what we call those B scans, one of these slices. Sometimes they're found in more than one. And then the idea is to try to get the algorithm to basically learn whether a volume has this or doesn't. Then basically they can flag it to the clinician. So we had such data on 1,200 volumes, and these are the percentages of. So we basically actually we had more, but we trained it on 1,200. And these are the percentages of essentially the frequencies of these different features. And you can see this is not a lot. Even if you have 1,200 and you go to 2%, there's not so many patients that will have this volume. So you start with a very sparse data. There's not so much data that you can work with. And again, as I mentioned, deep learning basically requires a lot of parameters that you need to learn. So how do we basically accommodate for this? So one way to accommodate for this is using something called transfer learning. In transfer learning, you take a model that was used for a different task in a different data set. So in these two examples here on the right, this is ImageNet, which is a very large database of pictures, and these are not medical images. These are the dogs and the cats and the cars and so on. So there's a huge number of images over there, and there's a huge number of architectures of models, deep learning models that have been trained on this. So this is one type of data set you can use. We can also use a more kind of related database. So in this, what you see here is a paper by Kermani et al. That basically looked at images, but not three-dimensional, just one image per patient. And they trained these images for, so this is about 100,000 of those. And they trained those in order to find specific features that are different than the features that we're looking at. So the basic idea, you take the model that was trained on the large data and then you tweak the model on the smaller data. So this is not something that we invented. This is a known way of working around the fact that you don't have as much data. But the problem is that this, as you can see, both ImageNet and this data that we get from Kermani et al., both of them are two-dimensional. These are not volumes, these are just images. And what we have is a three-dimensional picture of the, or three-dimensional volume of the retina. So how do you accommodate for this? And this is exactly where we basically came in with an approach to do this. The basic idea is that you start with a three-dimensional picture and you stack the images. So a three-dimensional picture is essentially a lot of two-dimensional pictures, and we just stack them on top of each other in the order that they came in the original three-dimensional volume. So schematically, this basically looks like this. You start with the tiled image. So now you started with the three-dimensional volume, then you tile the images one on top of the other. Then you put it into a network that basically just thinks about this as an image. So this is what we call ResNet here. This is using this transfer learning. But once you have this, you have to take into account the three-dimensional arrangement here. So two images that are close to each other represent something else, or they have a different relationship compared to two images that are very far away from each other. So this is what we call slice aggregation, which is basically this part that essentially takes multiple images that are next to each other, and we do some calculation for each of them, and then we basically just get to a smaller and smaller vectors. And these vectors in the end are basically getting into something which is relatively standard, something called fully connected net layers, where we basically connect all of these to the next layer, and we predict the biomarkers based on those. So the basic idea here is both the tiling and the fact that after we use the transfer learning network, we aggregate the slices using the three-dimensional structure. So we compared this approach to the state-of-the-art approach for volumes. So there's a couple of options of how to go about volumes. One approach that people have been using is a three-dimensional, it's called 3D CNN, three-dimensional convolutional neural networks. You can also use 2D CNN, but 2D CNNs are basically made really for images, and 3D CNNs are made for volumes. But you can see that in what you see here in the picture on the right, the top part basically shows you the precision recall area under the curve, and the bottom one shows you the raw curve area under the curve. In both cases, you see a substantial improvement when you're using our approach, which is called Sliver-net, compared to the state-of-the-art, which is the 3D or the 2D, and you can actually see that the 3D doesn't give you much more than just the 2D in many of the cases. So just to conclude, so we have a new deep learning architecture that can basically be used for transfer learning going from 2D to 3D, and that's especially important because you can imagine that there's a lot of there's many applications where you don't have that many 3D labeled data, but you might have a lot of 2D, especially if you think about ImageNet and others, but also medical images. Often you have many more 2D images. We showed the utility on AMD biomarker discoveries from the OCTs, but of course, this is not necessarily the only potential application. You can think about any 3D medical imaging. And then one important thing here is that it requires you do need to have some label data, but it doesn't have to be very large. Basically, what we show is that with a couple of 100, 200, 300, at least in these applications, we were able to get the same results or very similar results to using about 1,000, 1,200. The details, a lot of the details of what we have are in this paper that was published recently in Nature Digital Medicine, and I'm happy to take questions.
Well, thank you for sharing that, and congratulations on the recent publication. That's very exciting. I'm going to ask the first question, and I'm just curious, you mentioned there are other potential applications for using the same technique where you can leverage existing models to make up for having a smaller data set than you might normally want. Like, in your mind, what are the other environments that would be, other applications that would be interesting to pursue? Is there anything else in the eye that you would like to go after next that would build on this work?
Yeah, I mean, so I think that in principle, so this is not something that we showed in practice. So sometimes there's a difference between the theory and the practice, but in principle, there's nothing special about OCT that I can think of compared to any other three-dimensional imaging in medicine, where you don't have enough label data, which is often the case. So you can think about MRI or CT, right? We basically looking at three-dimensional images. In many cases, you just don't have that many data that is so samples that are labeled. Often, what people do is they look at segmentation problems, and in segmentation, even if you don't have that many samples, you can kind of think of every image as basically there's many pixels in the image, so you don't really it doesn't really translate one-to-one between the number of patients and the number of volumes in this case. But in this case, we looked at features that basically you have just one number, it's a 0 or 1, just binary for every volume. So you don't have any information per pixel. You have information per volume, and that's basically the challenge. So anything where you can take a volume and you say that patient has a tumor or that patient doesn't have a tumor, but you don't know where the tumor is, for example, right? So you just know if the patient has a tumor or not. If you can't get too many samples for this, you could still use this technique. in principle.
So that it seems like that's a target-rich environment for exploration. It looks like there is a question. So do you notice that diagnosis depends more on analysis of a plane, in your slices, or a volume?
Well, if it definitely a volume. And the reason that I know that is because if it's just the plane, then the 2D CNN would probably do the trick. The 2D CNN essentially doesn't assume that you have, in the example that I showed, we compared the 2D CNN, 3D CNN, and our approach. If it's all just in the image, you would see it in the 2D CNN, because that just assumes that it just takes into account the images. It doesn't take into account the volume. So you do get some signal from the images. You do get something, right? It's not that it was random, but you get a substantial improvement by looking at the volume.
Great, thank you. And I actually have a lot more questions, but I want to be sensitive to time. And maybe it looks like your colleague is stuck in clinic, but if he joins, we can ask some more questions later. For the moment, I want to turn to learning from Dr. Hatamizadeh about his work with Revere methodology. And so if you could share your screen and start the presentation, that would be great.
Sure, thanks, Joel. So let me share my screen and okay, I'm assuming that you can see my slides.
Yes.
Thank you.
Thank you.
Thank you so much. So today I'm going to present Revere, which is a collaborative effort between our laboratory at UCLA Computer Science Department and Jules Stein Eye Institute. So this is a deep learning based methodology and also a data set for semantic segmentation and quantitative analysis of retinal arteries and veins in infrared reflectance imaging. So I denote this as IR imaging for simplicity in the rest of this presentation. So as I mentioned, so this is a collaborative effort. First, I would like to thank my academic advisor, Professor Dimitri Terzopoulos, and our collaborators, Dr. Steven Schwartz and Dr. Hamid Hosseini, without whom none of this would have been possible. Also, we benefited from the efforts of many others who are listed in this slide. So as you all know, hypertension is a major public health issue. So every year it claims roughly about 9.4 million deaths around the globe. And chronic hypertension can progressively damage the retinal vessels. So this condition is actually leading to hypertensive retinopathy. So there has been some studies in the past indicating that by just tracking the retinal vascular structure, one can identify novel biomarkers for coronary heart disease risk stratification. And so based on this, we and other people, so we basically believe that we can actually find novel biomarkers by essentially assessing the condition of the retinal vascular changes. So given this hypothesis and the recent advances in both imaging and AI, we would like to actually create a framework that allows the discovery of such novel biomarkers. So in essence, what we have created in this collaborative effort is, I would call it a comprehensive framework for essentially retrospective analysis of hypertension-induced retinal vascular changes in IR imaging. So this has three components, and these components, each of these basically denote to one of the early signs of hypertensive retinopathy. So the first one is a vessel width tracking. So essentially the idea is that given an IR image, as shown in the left. So we would like to be able to segment them properly into arteries and veins. And given the segmentation maps, we would like to be able to extract pixel-wise width estimation maps. So we would like to know the diameter changes of these vessels. The other one, which is shown in the right, is artery and vein, or in short, AV crossing quantification. So as I'm showing here, essentially what we aim to do is to not only detect the AV crossing, which is a sign of hypertension, but also to assign a label regarding the state of the disease, whether that could be mild or severe, depending on the case itself. So in this work, I'm not actually going to talk about the AV crossing quantification, but just wanted to mention that that's part of what is essentially developed in this framework. And the last one is central light reflex quantification. And I can actually relate that to also the first one. So by actually segmenting the vessels and obtaining the maps, then we can also track the light reflex changes along the retinal vessels themselves. So here, as an example of use case of what we have actually created. So this is a case from our clinical team regarding the retrospective analysis of a patient's retinal vascular. So in this case, you can see that over the time, there is a focal dilation across along the retinal vessel. So this condition, which is also attributed as aneurysm, so this is also going to manifest itself in not only the changes in the light reflection, but also the change in the width of the vessel. So by tracking this patient over time, if our tool can actually be employed back in early days of the disease, as shown here, let's say in 2016, then we could essentially predict that in the future, this patient is going to develop such a condition. So briefly, before jumping into our methodology, I would like to mention that the reason we chose the IR retinal imaging is because it has many benefits in our case. So first, it's very comfortable for patients because, as you know, the IR wavelength is not part of the visible spectrum, and you can actually obtain both the image and video from the patients. Also, it's better or better suited to penetrate the media opacities for various conditions such as cataract or vitreous hemorrhage. And it's also easier to segment artery and veins using the IR images. And lastly, it comes with a measurement a scale on the OCT, basically images that from which we can find the width of the vessels and use that for our early predictive model. So here, essentially, I would like to summarize what this work has. So our work essentially produces a data set and also a methodology that allows us for segmenting the vessel, as you can see in the left, and also measuring the width of the segmented vessels. So this data comes with 46 retinal images. As I think Aaron had a very good, nice presentation delineating the fact that in deep learning, first, you need a lot of data and it's not easy to actually obtain these data. So this data set actually offers a valuable collection for creating deep learning based approaches for semantic segmentation. So the images, as you can see, they come with different retinal vascular conditions, such as retinal hemorrhage, vessel narrowing, AV leaking. And so we can actually exploit these images for building both a methodology for segmenting the vessels and also creating early predictive systems. And in comparison to other data sets, I list well-known or most commonly used data sets in our area. And as you can see, so first of all of these data sets are in the color fundus modality. So we are the first data set in the IR imaging modality. Second, except for one, none of them offer AV labels. And also the fact that our data set offers almost more data points than most of these data sets. So these are all the benefits of what we have in RAW-VIR dataset. So as I mentioned before, we actually propose a deep learning based methodology in our work. And again, Aaron had a very nice introduction on what deep learning is. So I will not try to actually go to the basics, but here we actually try to segment the vessels by employing an encoder-decoder framework. So one thing that is of particular interest here is that in addition to the main encoder-decoder that is used for segmenting the vessel, we propose to learn how to reconstruct the input images using a separate decoder. So this has many benefits as observed in our experiments. So first of all, it allows us to capture the fine-grained details of the segmented vessels, as I will show you some qualitative examples. So it basically allows us to capture these spatial and geometric details. And the second one is it acts as a regularizer, so our network would not overfit to the training data. So overall, this network architecture has been optimized specifically for this application. We also propose to supervise this network by a combination of dice and cross entropy loss functions. And additionally, for reconstruction of the input image, we basically use an L2 loss for basically reconstructing the image. Here also, we propose algorithm for essentially obtaining the width estimation maps given the segmentation maps. So, and the idea here is that, so we try to obtain a distance transform from the medial curve of the segmented vessels. So one good thing about this is that the clinician can actually interactively find the vessel width across any area in the image and be able to track it over different visits of the patient. So I think that this is a very useful functionality, which was first proposed actually by our clinical members in the team. We have also used very common metrics for evaluating and validating the effectiveness of our approach, as you can see here listed, most notable one being the accuracy and the F1 score in machine learning. And for evaluating the performance of our vessel width estimation, we actually employed the mean absolute percentage error, or in short, the MAPE metric. So for artery and vein segmentation, we showed that we outperform all the existing networks that are out there, specifically the UNet architecture, which has been used in many different applications. So we show the benefits of our methodology, not just in one metric, but over 5 metrics for different classes, artery and vein. On average, we also achieve the best F1 score. And for vessel width estimation, so first thing first, our approach as shown here qualitatively looks very similar to the reference maps, which are in the middle. So this is what our model outputs. And second, quantitatively, our approach achieves the best MAPE score among all the other methodologies using the same algorithm for generating the vessel width estimations. And here I have some qualitative comparisons of our model's output versus UNet and also the reference map. So first, you can see that our methodology can accurately segment both arteries and veins. So there are two other things that I would like to mention. First, you can notice that our approach can actually maintain segmentation with better structural consistency. So here the UNet actually alternates between the two classes in different branches, but our approach can actually maintain basically the class labels across different branches. And the second one is that it can also capture the details of the smaller vessels, which is often missing from the UNet segmentation outputs. So the last thing that I want to mention, and this also goes to one of the points that Aaron mentioned in his basically keynote, is the fact that, so in deep learning, basically obtaining data is not easy. And for this, we here show that using the networks that are pre-trained on our RAWVIR dataset, we can actually obtain state-of-the-art results on binary retinal vessel segmentation datasets for colored fundus images. So here we have two case studies, Chase DV1 and DRIVE. You can see that, for example, for Chase DV1, we can achieve a significantly higher F1 score in comparison to the closest methodology, which is Ethernet. And the same goes for DRIVE. So we show that the pre-training has its own advantages. And this is also confirmed by our qualitative comparisons. So our approach segmentation output is in the last row. So you can see that it's pretty similar to what the label should look like in the second row. And this is all coming from pre-training on our data set. So in conclusion, in this work, we presented a novel data set and methodology for semantic segmentation and quantitative analysis of retinal arteries and veins in IR imaging. And our method can actually segment both arteries and veins without any pre-processing or post-processing. And it can output very precise vessel pixel-wise width measurement maps. And we show that basically this work can enable quantitative and assessment of retinal vascular. So essentially you can study the morphological changes that are associated with retinal arteries and veins, and you can create early predicted models for diagnosis of various diseases in this area. And lastly, we showed the advantages of pre-training on our datasets for basically other domains such as color fundus imaging. So with this, I would like to end my presentation, and I would be happy to entertain any questions.
Great. Thank you very much for that presentation. So if the goal is to move these techniques into the clinic where doctors will be able to catch disease states as early as possible, certainly earlier than they do today, what are the next steps that you would like to see happen to make that happen, either in your own lab or with other researchers?
Sure, thank you. And that's a very good question. So essentially our RAWVIR framework enables studying these morphological changes. So the next step is actually a larger scale study that has retrospective data from patients that come from maybe different disease categories. And then we can put RAWVIR to test to essentially measure the changes in the width, also the light reflex, and also the AV crossings. So our framework is actually what that enables this study. So without this, it would be very hard to conduct such a study. So I would say that the next step would be just to utilize this. And the other thing is, so I think right now, this is somehow in a stage that can be directly used in clinic by like an ophthalmologist. So for example, the width estimation maps, so they can actually interactively look at the different areas in the vessels and see how it has changed throughout these different visits. And this can also be one benefit of what we offer in this framework.
That's great. So you're already at a stage where this isn't just in a research lab. This is something where you really want to start interacting with a hospital or even have a commercial entity conduct a study.
Correct. I think one of the, sorry to cut you off. So one of the advantages of collaborative efforts such as this one is that since we are doing this in with getting feedback from the ophthalmologist, so we are actually creating something that is practical. It's not just an early stage study in the lab, so it can be utilized directly in practice, because what we're doing here is based on their requests, so whatever need they see in day-to-day clinical needs. So I think, yes, I totally agree with you on that part.
Great, great. I have an audience question. Does patient retina color affect modeling or image analysis?
I think as long as we're doing the IR, it should not affect it. But if you're looking at a different modality, then that can be a factor.
Great, great. So I want to bring Dr. Halperin back into the discussion, and gosh, there's so many questions that I have about this. So I'm curious. I know that you and I have discussed that the Robert framework actually can take a model that's been trained using IR data, and it can be applied to other lightweight, data collected with different wavelengths. And then from Dr. Halperin, I'm hearing that there is this mechanism which is new, at least to me. I hadn't heard of it before, where you can figure out a way to use an existing network and train your data on it to get new usage. So I'm hearing these two different ways where you are able to leverage an existing network and apply it a different way. Is that common? How new is that? I'm interested in how we're overcoming problems with data, with labeled data. You hear data is the new gold, and I'm hearing two separate use cases where you're figuring out ways to utilize networks in ways that people weren't classically thinking of them. So I'm wondering if in your research you're figuring out more ways to get around these smaller data sets. And either one of you, both of you, can take this.
Yeah, I mean, I think the idea of using what has been done before and just adding on top of this is basically it's not only true for deep learning. I think it's true for any field of science. So even what Ali was presenting, the idea of the kind of a lot of the ideas are basically out there, and then you add on top of this some of your own ideas. That's very typical to any project in science. No, I mean, it's very rare. I can't really even think about one example that everything was started from scratch. And so the issue is that for deep learning and machine learning in general, you need data. And so it's maybe more kind of it's more clear that this is really what you need to do, because if so for example, Lee was mentioning UNet. UNet is a well-known architecture for segmentation, and that's been studied before on other types of data. So essentially, when you take the ideas, even if you don't take exactly UNet, you take it and you change it, you basically use all this other data indirectly. And so this is very, very common in all parts of deep learning, machine learning, and I think generally in science. You just add another layer to in every step that you do. I think what I was presenting, it was very explicit. We basically just took one part of our network, is just essentially taken as is, and then we add on top of this. What I think in the example that Ali showed, that this was a bit different, because it basically took some ideas and then they changed they didn't take any one specific block and just plug it in, but changed it. Even that block was essentially different. Great,
thank you. I like the reuse, recycle, repurpose pattern. That's encouraging, because we're trying to speed up how quickly this can get applied across a broad range of indications to improve the state of what's practiced in the clinic. I have an audience question. Let me.
I wanted to add to that, and I totally agree with Aaron's response. So I want to also mention that our application regarding segmentation requires some pixel level labels for arteries and veins. So that's why the importance of Ravi dataset comes into effect. So right now, there is nothing out there that you can utilize for this particular application. So I think that's why we at UCLA, we are in a very unique, basically, a stage or situation that we have access to this data through this collaborative effort, and we have created something that nobody has done yet. So I think the particular application that I just, but yes, for the application that Aaron actually mentioned, I'm very surprised because it was a smart way of utilizing the ImageNet for a medical application, and both are in the same realm of basically classification and detection, right? So I think both methods, they have their own merits, and they are innovative, and as nobody has done yet.
I was surprised as well, which it took me a while to ask my question, but I was surprised and found that interesting. For the Robert dataset, I know that you and I have spoken in the past about making some aspect of that data freely available for academic use. I haven't had time to follow up on you. Just for our current and future audience, is that true today, or should people be finding me to figure out how to license some subset of the data? Where are we at with making that available for other researchers?
Yeah, that's a very good point. I think we should discuss that later, but as in this current stage, it might be feasible to only release some portions of the training set for researchers to be able to prototype and see how basically it is done. But for real clinical use, I think it still requires the entire Robert data set that we have developed at UCLA.
Great, thank you. And yes, for people in the audience, if that's of interest to you, you can reach out to me and find me through the TDG at ucla.edu website. So the question from the audience is, how long do these models take to make a diagnosis when acquiring or analyzing new patient images? That's actually very interesting if someone's in the clinic. And the follow-on question is, do you imagine it will become faster as more data becomes available? Great question.
I mean, one of the nice things about deep learning is that it's very fast to, once you basically have a new patient. What takes time is the training. But that part is not done when you're basically, when you have a new patient. That part is done on the data sets that we've been discussing.
Yes, and to add to that, so we have two things called training and inference. So as basically Aaron mentioned, so training is done before, maybe it takes days, but that's not something that the patients or doctors need to care about. So eventually at the end, the inference is, well, the model actually takes some time to make a prediction. For our case, we actually use an approach called a sliding window. So we scan different portions of the image, and basically we try to output some probability predictions, and the entire thing can be done within 1 minute. And the reason that it takes 1 minute is because we have to actually do many passes of the same image to actually obtain reliable predictions for clinical usage. But I think it's still under a minute is something that can be totally utilized in clinical setting based on my conversation with ophthalmologists.
Great. And that's actually a lovely segue. Dr. Sada has been able to join us. Thank you, Dr. Sada. I know you're in clinic today, so really appreciate your time. We've had both presentations, and now we're discussing clinical application or clinical considerations for applying these algorithms in practice. And we just heard that being able to produce a result in under a minute should be something perfectly reasonable for using in the clinic. The follow-on question, which I'm curious what your thinking is on this, and this is from the audience. Do you imagine these models may eventually be used or be able to be used to diagnose at the home? So instead of going into clinic, the patient would be at the home doing something with the machine learning algorithm. Dr. Sada, do you have any thoughts about that? Oh, and I'm sorry, you're muted.
Yeah, sorry, I just unmuted. Hopefully you all can hear me. Yeah, no, absolutely, it's a great question, and I absolutely think there are potential applications there, especially since we are sort of in the era where we're developing now home diagnostics, even a home OCT device. is currently, that company is doing their pivotal trial for their home OCT device to get hopefully FDA clearance, but there are others. I'm aware of multiple. And so I think that's a shift we'll see in healthcare in general, is that there's going to be more and more home diagnostics to essentially, I think that's going to be a great path for how we're going to be reducing healthcare costs and the like in the future. But absolutely, I imagine that these kinds of the automated image analysis tools to go along and pair with those home diagnostics is going to be pretty vital.
And in both cases that we've talked about, there is this notion that you track someone over time so that you have early detection of disease states. Do you envision that this is something that a device that's in someone's home, and I'm trying to imagine in my head how it works with you as a clinician? Does it track them for years, and then at some point you get an alarm? What do you think that looks like when you're trying to get these?
Right. So the first application of home diagnostics is going to be in patients who are, we anticipate at least, in patients who have known disease, who then need to be treated at periodic intervals based on disease activity. So currently, those patients have to come in essentially on a regular basis, sometimes on a monthly basis, to be monitored and assessed in the clinic to determine, is the disease active? And if so, then they need to have a treatment administered. So you can imagine, if you have the ability to acquire the image at home and then automatically analyze it, then yes, exactly. The idea would be it would detect, maybe the patient could even scan themselves weekly then, theoretically, and the moment that activity is detected by the algorithm, then the alarm would come not only to the patient, but to potentially the physician, so they know that an appointment needs to be scheduled. So that would be how I'd anticipate it would be rolled out.
And would that also help titrate when medications are applied that might have side effects, so you can minimize side effects? Is that something that you see as a benefit?
Absolutely, because right now, the most common approach that's used, for example, to treat wet macular degeneration, which is the leading cause of blindness in the US, is that patients are treated with essentially treatments that have to be administered into the eye. And the way that's done right now is you treat a patient. If there's no signs of activity, when they come back, you treat them again, but then you wait for a longer interval till they come back. But the problem is you're treating the patient even when they don't have activity, in part because you're worried about missing activity that might develop in the interval. So if you have a reliable home diagnostic, then you can essentially treat the patient only as needed, exactly at the time they need to be treated. And that might minimize some of the toxicities that these treatments have by administering only the minimum number that they need.
Fantastic. This is very exciting. So Dr. Sato, you jumped on just in time. We're actually now, well, actually, we might be able to squeeze in one more question and then we should wrap up. This is from the audience, and I want to thank my colleague, Ricky Rath, who has been surfacing these questions for me. As imaging methods improve, does image resolution or image size have more impact on developing better diagnosis? I don't know if anyone wants to take that.
Well, I can take the first stab at it. I think that, quite frankly, in at least at the present time in clinical diagnosis and ophthalmology, at least, which is what I can speak about, we're doing pretty well on the resolution side. We probably could benefit from tools that give us great contrast. That's actually been one of the exciting things about the work that's happened in deep learning as applied to ophthalmology. Is sometimes there are features hidden and coded in the images that we're not even aware of, that actually seem to be of predictive value. That's actually been a revelation recently in imaging. So, I mean, again, is there even more that could be discovered if we had even higher resolution images? It's possible. But right now, we haven't even fully realized what we have with our current diagnostic technology. So that's what I would say at the present time.
Maybe just quick comment related to this. So, when we look at the volumes that I mentioned for the OCT, we looked at I think it was 97 images in each of them, but in many cases, the ophthalmologists don't take 97, they take just maybe 20 or 30, and Dr. Sato would know exactly the number, but basically, we were able to show that you don't really need the entire 97 to get to the same results as we got. So it would be enough to look at around 30, let's say, which is kind of resolution in one of the axes, if you think about it. So you have each of the 2D images, that the resolution of these, and then basically the number of images. So at least in one of the axes, we showed that going to about half or 1/3 of the images would give you enough resolution to do at least these tasks.
Great, great.
And I also wanted to add that I think our data sets are sufficiently large in terms of the image resolution. And I agree with the notion that at this current stage, it's an exciting time to exploit our latest advances in imaging technologies and be able to directly apply the latest and the greatest AI methods. I don't think that we are still in the threshold of what AI can achieve. There is still so much to be discovered, and the methodologies that we discussed today, I think there are some great ways of getting into this.
Great. Well, so we're past the hour. So thank you all for talking about all of these discoveries. In my mind, we're now at the step where what we really need is partners for clinical studies and partners for commercialization. And in my own mind, I have this vision now where we're all going to get, or the people in need will have these devices at home, and they'll start tracking early stages of disease, whether it's hypertension, everything that hypertension can cause, whether it's macular degeneration, and I'm sure you all know better than I other diseases of the eye or other disease states that can be detected through the eye. So I'm excited that now it's on TDG to help find those partners to get this out into the world. So thank you again for sharing, and look forward to using this talk to get the word out to further all of that clinical collaboration and licensing to commercialization. So thank you all very much for your time.
Thank you.
Thank you very much.
Thank you.